All right, so let's start here. How's the sound? Everybody hear me in the back? Everything's cool? Yeah? Okay. So, did everybody have a good weekend? Rested, recovered, ready for more tests? No, me neither. All right, so let's get ready and let's focus here because I know we are okay, guys. Let's All right, I know we are a bit behind. Hello. Should I do a little dance here? All right, everybody, let's get serious. Is everybody focused? Yes. We're here for immunology. Yes. Tough concepts, not intuitive. Right, so pay attention. Okay, I know we're a bit behind with these topics, but I really always prefer to cement the fabric of what are the fundamentals for those immunology principles rather than just rush through the material and tell you good luck, right? I don't like that. But especially with these parts that are so important to get on what is happening with interactions, what is the reaction or the action of a particular lymphocyte. What is the use of having these lymphocytes going all over the place? And what is really happening at the lymph nodes, the secondary lymphoid tissues? And how is it that you are going to mount a good immune response and the factors that are important to mounting out that response? Those are all very, very important fundamental topics that a lot of times, although you read about it, it doesn't really stick to your brain. Well, at least it did not stick to my brain for a long time. And every time I look at it, if I stay without looking at it for a long time, it's like I'm looking at it for the first time. It's kind of weird. Immunology can do that to you. Okay? So let's focus here on the parts from where we left off last lecture. Okay? And what I will do is, for those topics that are actually easy to grasp, I will make the supplemental videos. I will add the supplemental videos because then I know that you can navigate through it without a lot of issues. And sorry, but that door is still driving me crazy, so I'm gonna leave it open. It's pronounced slam every time, all right? Okay, so we left it this here. Now, we left it at this slide saying that in order for a lymphocyte to activate, again, think about this we're looking now at the T cell, but this also applies to the B cell. Okay, in order for a lymphocyte to activate, they need to get at least 2 signals. Okay, so here it says it requires 2 signals, but that's the bare minimum. They require at least 2 signals. Usually 3 signals are good enough. But in order to have a very good activation, a very good response, you're going to need to really stimulate those cells to do their jobs. Okay, so what is happening now? The first signal that you're going to have for this T cell is going to be recognition of the peptide that is meant to recognize. So remember that when you made that T cell receptor, where is it again you made the T cell receptor? In the thymus, right? When you made a T cell receptor, you made it specific for an antigen that is not self, right? So it will be able to look at whatever the MHC molecule is presenting, and it's going to be able to recognize it, but it has to be not self. So let's see here. If you have an antigen presenting cell that has something on the surface, and one T cell recognizes it, and that's the only thing that happens, then after that, nothing really is going to happen to the T cell. Okay, so one recognition, which is the first recognition, which is the stretched out protein on the MHC, only that, and the T cell, it's like, oh, it's not going to do anything. It becomes anergic. Okay, so only one signal is a signal for the T cell to become anergic. It's just going to be hanging around just like a zombie. Okay, you can talk to it and it's gonna go, hey zombie, what's up? Nothing is gonna happen. And then it's gonna die, because zombies, they're already dead. But in this case, it will die later, okay? All right, so this one signal type of signaling is part of tolerance. Okay, it's part of your immune system to really make sure that your T cells are not going to go crazy just because they see the antigen and nothing else. Right? So it's a part of what you call peripheral tolerance, because it's not happening in your primary organs, primary lymphoid organs. Okay, so now let's see what happens here. Okay, so this is blah, blah, blah. Okay, so energy. Okay. Okay. So now we're going to start getting serious about this co-stimulation of the T cell. All right, so first thing that's going to happen here, you are still going to have the recognition of that little peptide, right? But now this is when we see the usefulness and the function of your professional antigen presenting cells, which are mainly those dendritic cells. Okay, so these professional antigen presenting cells, when they are presenting a piece of the pathogen to your T cell, they also come with other ligands on their surface, specifically this B7 ligand. And this ligand is going to interact with the particular receptor on the T cell, that is the CD28 receptor. Okay, and I think I mentioned before that the B7 ligand is also called CD80, CD86. Okay, but we just call it B7 to make it simple. It worked. It worked again. Okay, so B7, just to make it simple. Okay, I almost did it. So what is going? So let's pay attention here. Professional antigen presenting cells, they come with B7 on their surface. Okay, they didn't have B7 all the time, but they started making a lot of B7 once they had recognized that pathogen all the way back in the tissue where it picked it up. Okay, so they are very good at expressing a lot of B7 or just enough to stimulate the T cell. Okay, so here you have your first signal, and then here you're going to have the interaction for your second signal. Okay, and in this case here, okay, let me not get too complicated now, but let's think your B7 CD28 signal is a very important second signal that is going to tell your naive T cell to not be so naive. It's going to start activating it. Okay? All right, so let's see what happens here. This I modify a little bit on the slides, trying to put wording that's going to make a lot more sense. Okay? So here, first thing that happened, you are going to have this interaction. Okay? Once that interaction takes place, you are also going to the professional antigen presenting cell is going to feel that signal and is going to start to secrete cytokines that at first are general to stimulate proliferation of your T cell. Okay, so one very important cytokine that is secreted at the time is interleukin-2. Okay, so your APC produces a little bit of interleukin-2. No, sorry, delete that. Is interleukin-12. Okay, interleukin-12. Okay, we're going to see more of these interleukin later. Interleukin-12 secretes a little bit of interleukin-12, is going to start to give a bit of a stimulus for your T cell to start doing something. Okay, after this secretion of interleukin-12, then you're going to get your T cell. All right, so make sure you really caught that, okay? You're going to get your T cell that had at least two signals. You're going to get this T cell to start acting differently. First interleukin that the T cell is going to produce is going to be your interleukin-2. Okay? And yes, and this is another reason why you're taking your next test in pairs, right? I hope everybody is studying in pairs, right? Or in groups, right? Because this stuff can be very, very confusing and the numbers are very similar. All right, so at least when you think together, you should get all the right answers. Yes. At least 2. Most of the time, 3 or more. But at least 2. Yeah. First one, what is your first signal? First signal is the T cell recognition of antigen. That's your first signal. Your second signal is your very important B7 CD28 stimulation. Okay, and now you're going to have a lot of co-stimulatory effects that are going to go on here. Okay, once your APC feels that this thing worked, it is going to start producing cytokines. One of the first cytokines produced by APC is interleukin 12. Interleukin 12 hits the receptor on the T cell. Okay, the receptors on the T cell here on the side for interleukin 12 are going to send a signal to the T cell nucleus to tell it to start producing interleukin 2. Okay, guys, let's be quiet here. Hello. What is interleukin 2? It's a very important cytokine produced by the T cell. T cells, especially helper T cells, all right, take that down, that's important. Helper T cells are very good at producing interleukin 2. Now, what is interleukin 2 going to do? Look at this, the cell that produced it has receptors on its own surface for that cytokine. So the cell that produced it is going to feel the cytokine that it produced. That is called an autocrine type of response. Okay, so the cell produces it and it feels it. So this signal that is going to come from the receptor for interleukin 2, this signal is going to tell the T cell to start dividing. Okay, so interleukin 2 is going to tell the cell, look, it's time to make the clonal expansion. It is really time to produce a lot of these specific T cells that could recognize that piece of pathogen. Because now you have to build up your whole army to go back to the site of infection and eliminate the pathogen. All right, so interleukin 2, very important for proliferation of the cell that recognize the pathogen. And how many of those cells are going to have around? The naive cells that recognize that one little pathogen? Well, not many. 1 or 2, 3 maybe in your whole body. Very, very little numbers, okay? Even if one T cell recognizes different antigens in the pathogen, the T cell that recognizes a specific piece of the pathogen of the antigen is very unique. The characteristic is very unique for this, like one cell, okay? Because remember all that recombination of genes that is happening in the thymus and all what the cell has to go through, right? So you have very, very small number of cells that recognize this one pathogen. So now you have to build them up. You have to build up your army. So now your cells are going to do this autocrine signaling. They're going to start to divide and they're ready to become effector cells. Okay? Everybody got that? Yes? All right. Yeah.
So only the dendritic cells upregulate the B7?
Not exactly, but they're the best ones at doing it. There are other antigen presenting cells that also have B7, but they don't have it to the degree that the dendritic cells have. So there are other cells that can start to push the T cell to differentiate, but they're not very good at it. Okay? Okay. So now other things are going to happen here. Okay? So the first thing that happened was here, recognition of your antigen. Now, when this thing recognized, that send a signal to the T cell, right? Well, but there is a little mistake here. So make sure you correct the mistake, right? Because remember, if you have only one signal, nothing else happens. That's energy. So you also had interaction here, which I'm going to put it on the side. Bless you. Bless you. Okay. So you also had the interaction here on the side of your CD28 with your B7, right? So you already had the second signal. Okay, so your T cell now is going to start doing things that zombies don't do. Okay, so first thing that it's going to do is going to start to upregulate this receptor, this ligand right here. It's called CD40 ligand. Okay, it's going to start upregulating this ligand that is going to interact with a receptor on your antigen presenting cell. Okay, and once this interaction happens, now your T cell is going to tell your dendritic cell that it has to do something else. Okay, so what is going to happen here? This interaction here tells the dendritic cell to make even more B7 ligands on its surface. So now you're going to have a lot of those B7 guys, even more than you had before. Okay. Besides that, the dendritic cell is going to produce more cytokines that are going to be important for signaling of your T cell, besides that initial interleukin-12 cytokine. Okay, which cytokines are going to be produced next really depends on what kind of pathogen is being presented. Okay, so you have all different soups of cytokines that can be produced by your dendritic cell. This type of co-stimulation is going to enhance how effective your T cells are at eliminating the pathogen. Okay, so what we mentioned here so far, interactions, of course, T cell receptor with MHC, B7 with CD28, and now CD40 ligand with the CD40 receptor. Okay, so this is important for your dendritic cell, and is also going to be important for another type of cell that we're going to see later. Okay, but very, very important forms of interactions. All right, so here comes your question. I don't remember how many questions I've put it in this PowerPoint, but you have at least a couple of points that you can get, minimal. Okay, what is the ligand for B7? Okay, what is your ligand for B7?
Okay, that's why I'm like literally.
Okay, 10 seconds. Okay, what is your other name for your B7? The other name, B7 has 2 other names. Okay, I'm gonna have to stop it. You're gonna get a good answer here. Okay, I was giving you a little more time to put the right answer in. Okay, so what's your answer? That is CD28. Okay, CD28 is the ligand for your B7. Okay, what is the other name for B7? CD80 / CD86. Okay, that's the other name for a B7. So of course A is not correct. What is CD4? The molecule on your T cell receptor that is going to recognize MHC class 2. What is CD3? On your T cell receptor that is characteristic of T cell identity. All right. Okay, so here, recap what's going on. Antigen presenting cell ate up the pathogen presented here to your T cell, right? And now you are making that T cell not naive, and you have your second signal. You're also going to have signal from cytokines, and you're going to get your T cell to really become powerful and make this whole army of clonal expansion that is going to go back to the site of the infection to kill the pathogen. So now we're going to start talking about how it does that. How does it go back to the site of infection? And why doesn't it just stay there in the first place, right? So what's going on here? You can read this on your own, okay? So this is what's going on. When you have T cells, right, that are naive, not mature but naive, circulating in your blood, what is it that they do? They're always patrolling. They have to go around the tissues, the organs, okay? And then they have to look for secondary lymphoid organs that might have a pathogen to present to them. Okay, so what happens here? If you have T cells that go in the blood, they get to the liver, they get to the skin, they get to kidneys. So in this case, they can also come out of the circulation. They can get to the lymph, that is interstitial fluid, right? And then they penetrate the lymphatic vessels, and they are going to circulate here in the lymphatic vessels, and then they're going to find here a lymph node, right? And in this case here, they're going to look for pathogens. If they don't find a pathogen, they're going to come out of the lymph node, and they're going to stay in the lymphatic circulation to meet another lymph node that is connected to this first one. So another lymph node is missing here. Okay, so here is another lymph node for you. Okay, so this guy is going to circulate to another lymph node. It's going to go through several lymph nodes until it doesn't have any more lymph node to go through, and then it's going to be dumped from this thoracic duct. Is going to be dumped by the very close to the vena cava, where it's going to go back to the right atrium and join here your blood circulation. Okay. Now let's say that instead of going to these organs, your T cell went to the gut circulation. Okay, it can come out of there, but now instead of going to your lymph nodes, your gut immune system has a bit of different arrangement in your secondary lymphoid tissues. So this arrangement is really called Peyer's patches, okay? And you're going to see later what is the difference between these secondary lymphoid tissues. Now, in this case, from the gut, these cells can go to Peyer's patches, check out if they are pathogens. If they are not, then they can rejoin the lymph and hop into the lymphatic circulation again, and they just keep on going around, okay? So you always have a lymph node that is going to be filtering all this lymph that is coming from this interstitial fluid. In the case of the gut, you're going to have those Peyer's patches, okay? So that's the story with your lymph. And then there is another story with your blood. So you have your T cells that are in your blood, they are circulating in the blood, they are not coming out of the blood, and then what they're going to do is that they are going to go and travel through the spleen. In the spleen, they are going to check out if there are pathogens in the blood. So the spleen is going to be the meeting point, the meeting place for presenting cells and pathogens that are blood-borne. So here is also filtering, okay? And it's the same type of filtering that you're going to find in your lymph node, but this is going to filter the blood, okay? And if this cell doesn't find anything, it's going to go back to the blood circulation, and it's going to keep on recirculating. It's just going to always look for a pathogen that it can match. And why is it that the T cell is doing that? Well, basically because, remember, you have very few number of T cells that can recognize one particular pathogen. And if that T cell stays, sitting in one place, it is useless because the pathogen can go anywhere in the body. So the T cell has to go around to look for, to be patrolling whatever can be entering from any other part. Okay, it cannot just be sitting around and waiting. Okay. So when you get your lymph nodes, Okay, so these are not pair patches. When you get your lymph nodes, there are two ways in which your T cells can hop in the lymph nodes. So here, the first way is through your lymphatic circulation. So the lymphatic vessel that arrives to your lymph node, that is called your afferent lymphatic vessel. Okay, and here is showing just one, but you actually have like several afferent lymphatic vessels. Okay, so you have several lymphatic vessels that can reach your lymph node. Now, if it goes this way, so fine, it's going through your lymphatic circulation. But it can also come from the blood circulation, because remember, lymph nodes are organs, and they need to be supplied with blood, with nutrients, with oxygen, right? They need to get rid of their byproducts. So they do have a blood supply that ends up in very fine capillaries, where you have your arterial capillaries. And then you have all this exchange of things that go on, right, in your capillary bed. Everybody took AMP, right? Are you taking it now? Yeah, you know what I'm talking about? Capillary bed exchange? Okay, who has never heard of capillary bed? Okay, so just picture in your head. Okay, picture in your head, you have arteries and you have veins. The arteries are carrying the blood that is oxygenated from your heart to your organs. When it gets to the organ, that artery is going to start to branch into very, very tiny vessels, and those are called capillaries, right? And only from the capillaries that you're going to be able to receive oxygen to maintain your tissues alive, right? And you're going to get nutrients from the blood, and also the cells that have already utilized the nutrients is going to put byproducts out, and things like carbon dioxide that you have to get rid of from the tissues, otherwise your tissue is going to die, right? So in the capillary bed, you have a lot of these very, very fine vessels that do all this exchange, okay, that is happening here. This is the capillary bed. And then right after that, you're going to have little venules that are going to join and make bigger veins, and this deoxygenated blood is going to make its way back to the heart. Okay, does that make sense? Yes. Okay, so picture now that this lymph node right there is getting the blood that is oxygenated. It has this capillary bed, everything is going on over there. And now, things that happen here in the capillary bed that are useful for cell function, the exchange here is only of small things. Cells do not go through that, okay? You're only going to have little pieces of proteins. You're going to have gases, gas exchange, right? Some sugars, right? But real cells, even the tiniest cell, like a red blood cell, is not going to go through that. Okay, but at the lymph node, you have something else happening. When you look at the venous side of the capillary bed, those little venules have different structure than everywhere else in the body. So they are only having this structure at your lymph nodes. Okay, the structure present there makes it possible for the T cells to come out of your blood circulation and sneak into your lymph node itself, inside the lymph node. So when you say, well, cells are never going to come out of the blood circulation unless you have a rupture, well, you always have exceptions, okay? So looking here at your lymph node, you're going to have specialized cells that are going to allow T cells to go through and reach the lymph node. So that tells you that the road of T cell to the lymph node has two paths. It can come through the lymph, right? Or it can come through little venules, okay? Straight from the blood. So two ways to get there. That's why you have this picture here. You can get there by train. I don't know why lymph node goes through train, or you can get there by car. Very simplistic way of describing two ways to get there. Modes of transport, right? Okay. So we're going to look more into the characteristics of this high endothelial venues. That's what you formally call them. Okay? All right. So everything important here is mentioned. Okay? So here we go. With those guys with the high endothelial venues, that's a difficult concept to grasp. Okay? In my head, never made sense until I finally figure out, oh, it's because of the capillary bed circulation. And then it started to make sense. Okay, so what is happening here? You have blood that is coming to the lymph node. So you have arteries here. And then here, look strategically where? Here, you have a certain region in your lymph node that you have those capillary beds, right? You have capillary beds everywhere else because your whole lymph node needs supplies from the blood. But specifically in this area here that you call the precortex area. Specifically in this area, you're going to find those guys that are called high endothelial venules. Okay? Little blood vessels on the vein side. Okay, so deoxygenated blood is starting to get there. Okay, it's starting to be produced over there. Okay, so when you look at these little capillaries, this is a blood vessel, okay, and you look at the cells, at the endothelial cells that make the wall of the capillaries. Do you know how many layers of cells make the wall of capillaries? One. Very, very tiny, one delicate layer of cells, right? So this one layer of cells of these venules, they are not made of the same cells that all the other capillaries are made up of. They are made of these strange cells that are kind of tall. Instead of being like a fried egg, they are quite tall. They look like a cube for something. They're very, very tall. So then some immunologists with a good sense of humor, they call them high because they are tall endothelial venules. Okay, so that's the whole thing with this thing, high endothelial venues. So what is happening over there? Over here, these high endothelial venues, when needed here, they have some specific properties that allow your T cells just to hop out of the circulation and into your lymph node. Okay. So here you have your naive T cell. Specifically, the naive T cell is going to go around because it's looking for antigen. When you have an activated T cell, then the story is different. Okay? The activated T cell doesn't need to be circulating through all this stuff to look for stuff. It already found it. The activated T cell has the job to go back to the site of infection and kill the pathogen. So it has to find different avenues to get the job done. Okay? So let me see what is important to know here that I have not mentioned. Okay. Okay, so how do you get to the lymph node? You can come from lymphatic vessels or you can come from the blood. Okay, so in this case here is telling you when your T cell gets to a lymph node and it doesn't find a professional antigen presenting cell, it has no business to stay in the lymph node. So it's going to hop into the efferent lymphatic vessel, which is usually one or two, mainly one. Okay. It's going to hop into efferent lymphatic vessel and it's going to travel to another lymph node and it's still going to look for pathogen. Now look what happens in this lymph node over here. Okay, in this lymph node, a dendritic cell that was in the tissue somewhere, it hopped in the lymph and it came to the lymph node and is going to sit there and wait for the specific T cell to show up. Okay, so the dendritic cell is not going to be circulating all over. It's going to go to the nearest lymph node, nearest to the site of infection. It's going to sit there and it's going to wait for T cell to show up. Now, let's say the T cell showed up and you recognize the antigen. This is where all the first signal, second signal took place. Now you're going to transform your naive cell into your effector cell. That effector cell is going to hop out of that lymph node and is going to search for the site of infection. Okay, so it's going to hop out this way and it's going to circulate everywhere until it finds a blood vessel that is very close to the site of infection, and we're going to see how that happens. It's going to find the blood vessel, it's going to interact with the cells here, with the endothelial cells of the blood vessel, is going to come out of the blood vessel and is going to attack the pathogen where it got in the body in the first place. Okay? So now you can imagine all the type of molecular interactions that are going to tell this T cell, you know, come out here close to the skin and do not come out close to the pancreas. How does it know where to go? Right? It's kind of complicated. How does it find out? Okay? So here, what is happening here? This is when you have a lot more of these weird names that are important molecular interactions. Okay? So here comes T cell from your blood. It gets into those guys, high endothelial venues. Okay? So they come out. They enter the lymph node at the T cell zone, because that's where T cells like to hang out, paracortical space, right? And here it is going to interact, find the, get activated, come out, and go look for trouble, where the trouble is. Now, how is it that is coming out of the blood and getting into the lymph node? That's when all these numbers and letters start to come up. Okay. First thing that's going to happen, your T cell that is not activated, so this using color code, red for not activated, greenish for activated. This guy is not activated. What does it have on the surface? Look at this. It has CCR7, which is a receptor for interleukin 7, or for other also different things. Okay, it has CCR7. Not only can interact with IL-7, it can also interact with receptor or ligands at the high endothelial venue, right here. It has L-selectin. Remember we had seen before when we started looking at rolling and extravasation. Remember that lecture? Okay. It has L-selectin. It has a T cell receptor, right? So what's going to happen here? At first thing, when it gets to the high endothelial venue, it's going to start weakly interacting with CCL19 and CCL21. Okay? Those CCL19 and CCL21 are on the surface of those tall endothelial cells. Okay? So this guy here is going to be CCL19, CCL21. CCR7 is going to interact with them, and you're going to start rolling on the surface of that high endothelial venue. So remember, rolling is like, okay, I'm sticking, but I'm not totally sticking. I'm just passing by. Okay. After that, you're going to have a stronger interaction of L-selectin with the PNAD. That's not the only thing that interacts with. Okay. But this is what's going to give you one of the stronger stop signals. Okay, so first it's rolling and then it's going to stop. Those signals are going to tell the T cell to squeeze through those high endothelial venues, cells, and get inside of the lymph node. I do have a better picture for this. Okay. Okay, we will get to the better picture for this. Okay, so the bigger picture of what is happening here. There was one site of infection, right? Like, for example, they're showing here the person stepped on a rusty nail, and I do have the link for this video, which is very interesting because it happens all the time. Like when a person is going to step on a nail, it has to be rusty, right? If it's not rusty, it's not going to cause trouble. So step on the rusty nail. Let me tell you a little, almost like a joke of what happened to my neighbor, right? Because you remember last time we had a hurricane, a serious hurricane, and a lot of fences went down and all this stuff. So my neighbor has a cat that wanted to adventure out of the house right after the hurricane, and all the fences are down, and she can't really see because everything is wet. So the cat sees another cat on the other backyard, which he could not see before, of course, because there was a fence there before, right? So anyway, he comes out and he's like gonna charge at the other cat. So my neighbor comes out and goes to grab him, but of course she stepped on a rusty nail, right? So then she called and said, what should I do? Should I go get a tetanus shot? Should you get a tetanus shot if you step on a rusty nail? Yes, you should always get a tetanus booster shot. Because as we're going to see, your immunity against tetanus is not as good as a T-mediated immunity. It's not a T-cell mediated immunity. Anyway, we're gonna get to that. So what's happening? So she calls me and then she says, I said, go get a booster shot. Okay. So she goes to get a booster shot. And then I'm telling my husband, you know, Carmen, my neighbor, she stepped on a rusty nail, and then he says, how is he? I said, oh, she is fine. He goes, no, how is Rusty? Because it so happens that Rusty is the name of the cat. So my husband thought that she stepped on Rusty. He's like, how is he? Still alive? No, and they're like, oh, yeah, okay, Rusty. So she did not step on Rusty. She stepped on the Rusty nail, and she's fine. She got the booster shot. Nothing happened. Okay. So after you stepped on the Rusty nail, what's going to happen? You're going to have here some bacteria. They're going to be, or toxins, whatever that is. That's going to be picked up and carried by those dendritic cells, right? Remember, first they phagocytose and then they move around. They become non-phagocytic. They go to the nearest lymph node. They don't just go around to another lymph node because that's a waste of traveling time. They go to the nearest lymph node, which is also called the proximal lymph node. Proximal to what? Well, to the site of damage. Okay? So they go to the first one that they can find, and they're going to sit there, and they're going to wait for the T cell to show up. And the T cell is going to show up because the T cell is circulating. Okay? Now, imagine that if your dendritic cell gets there, doesn't find the T cell, and gets out of there and go to the next lymph node looking for that T cell to match, you're going to have a bigger problem. Because your T cell matching this one is going to be traveling in the circulation looking for that matching dendritic cell, and by the time it gets here, your dendritic cell is going to be over there. And it's not like you're going to stop your circulation. So if the dendritic cell doesn't sit around and wait, you're never going to catch up with it. Does it make sense? Okay, so when you're going through this and you say, oh, the circulating T cell reaches the lymph node, the dendritic cell is there. Wait, instead of going through it and reading, and it's like, what are they talking about, right? Get the big picture. Dendritic cell, carry the thing to the nearest meeting place, and it's set there and is waiting for its date to show up. If it keeps on traveling, it's never going to meet the date, okay? Then the T cell comes in there, meets the dendritic cell, and they live happily ever after. They're going to do their thing. They're going to activate first, activate second. You know, they're really going to hit each other, right? Just a little glance is not enough, right? A little glance and they become zombies. Okay, so that's happening at the lymph node, right? And let's see here, important to know about that interaction. Here we go. Ready? Okay, important thing to know about this. How is it that your T cell is going to get there? It's going to come through the afferent lymphatic vessel. And here you're showing two of them because you have more lymphatic, more afferent than efferent. Afferent for arrival, okay? It gets in there. It goes to the zone where you have the T cell hanging around. Okay, so here you're going to have, okay, the yellow one is your T cell zone. Okay, so the T cell zone is saying like, well, is it, am I going to find my match here? Am I going to be activated or not? So let's see what is happening next. Again, the T cell could get there either from the afferent lymphatic, or it could get here from the high endothelial venue. Anyway, it ends up in the T cell zone. So here is a story with your high endothelial venue again, okay? So you do have your CCR receptor on your T cell, and they're going to be interacting with CCL19, 21, or 21. Okay, we'll put them together here, CCL19, CCL21. And how is it going to know that behind this high endothelial venue, you're going to have your lymph node? Because remember, this thing doesn't see anything. It's just in the blood circulation. And it doesn't know what is behind that blood vessel. Okay, it has no idea. What it's going to tell it is that the amount of those binding molecules that are at those high endothelial venues, the number of those molecules for this thing to roll, that is going to go start to increase. So the cell is going to be rolling and rolling, but then it's going to start to roll slower. Okay, it's going to roll not as fast because it knows that something is happening there. When you start to find a lot of CCL19 and CCL21, you know that something is going to happen around there. Okay, so high frequency or a gradient of CCL19 and CCL21 is really telling your T cell to roll around and watch out for better interactions. Okay? Now, when that happens and you actually find the place for like right inside the lymph node, then you're going to have this other interaction of L-selectin. Let me see here. Next one. Okay, these are all the other interactions that are happening for the rolling. Okay, higher binding due to frequency, but I want to get to the one that's really important. Okay, this one right here. Okay, you have interactions CCR7, etal, CCL19, 21. You also have interaction of L-selectin with addressing molecules, and we have seen that before, right? When those things are happening in high frequency, you are going to have increased production of that guy, LFA1, in your T cell. Okay? LFA1 is going to be able to interact with that very important addressing that is called ICAM1. Remember what ICAM1 is intended for? Extravasation. It's like, it is not the slow down stop. The slow down step is for rolling, CCL19, 21. This guy, ICAM1, is like, stop. Don't roll anymore, just stop. So ICAM1 interaction is the stop signal. So the cell is going to be rolling and rolling, and when it gets there, like, I'm stuck. That's what it's supposed to be. It's going to stop. Now, once it's stopped, then it's going to start to extravasate out of the high endothelial venue to get your lymph node. Okay? So those are important things that are happening inside that high endothelial venue. So this is just to recap what I just said, okay, including the interaction of those molecules that are so important, okay? And you can read through this. I just mentioned that, okay? All right. So once it gets into the lymph node, it's going to look for the dendritic cell that might be presenting the antigen. So is everybody cool with that? Okay. Yeah.
So that whole process is just for them to get into the HEV?
So that they will get out of the HEV and get into the lymph node. Because if they stay in the vessel's blood circulation, they're never going to reach the lymph node. Okay, so they have to come out of there. And the capillaries do not allow, regular capillaries do not allow cells to come out of there, of the blood circulation. They only allow little molecules and gases.
So that's like water.
Yeah, they will get to the lymph node from the blood. Okay? So here's the whole story on what is happening here. Okay, so the same thing as I said before. You have your dendritic cell that arrived at the proximal node, right, the nearest lymph node, and is going to hang around strategically in a place where the T cells like to hang around. So that is going to be your peripheral region. Is it written here? No. It just says T cells on, the one in yellow, okay? So it hangs around and it waits for that T cell to show up. T cell shows up, interacts with your dendritic cell, and this is where you have that activation. What is going to happen? The first interleukin that your dendritic cell is going to show, is going to put in there. Interleukin 12, right? Then you're going to have your co-stimulation, and you're going to have your T cell to produce interleukin 2. That's your T helper cell. Helper cells are the good ones to produce interleukin 2. Keep that in the back of your mind because that's important. Okay?
So for the helper cells, is that, do they only have the CD4?
Helper cells have CD4, yes.
So no CD8?
No. Helper cells have CD4, T cell receptor, CD3, and all the other stuff, the T cell receptor. Only the cytotoxic T cells have CD8. Okay? All right, so those are your helper cells. We're going to find more later more about those helper cells. Okay, so once you have your clonal expansion, these guys are going to lose something on their surface and gain something else because now their job is different than it was when they were naive. Okay, so they're going to leave. They're going to leave that. Now, besides those addressing interactions that I had already mentioned, right? We have to think, well, when the T cell is in the lymph node, what is it going to make it come out? It can just stay there and hang around there, right? Because we know that when it came to the lymph node, from the blood, for example, it had those kinds of interactions. When it came to the lymph node from the lymph, it kind of was just filtered there, right? Now, what is going to tell the T cell not to hang around the lymph node and to circulate again? So this is the story where this S1P is not CYP, okay, is S1P, okay, that comes along. Okay, so what is happening here? What is this mechanism here? Here you have your blood circulation, so those are arteries, okay? They're going to get to your capillary bed and going to get to your higher endothelial venule. Now, what is happening here? In these arteries, on the surface of those endothelial cells, you have a molecule that is called sphingosine 1-phosphate. Sphingosine is a type of a sugar molecule, okay? So it's on the surface of the cell. It's called sphingosine 1-phosphate. So we abbreviate that as S1P, okay? As you are getting to those close to those high endothelial venules, the expression of this molecule starts to be a lot, okay? So that means for your blood vessels that were before, you did not have a lot of expression of the S1P. You had a little bit here, a little bit there, and then you started having more and more expression until it became a lot of expression, okay? So how is that important for the T cell? So here comes your T cell. Here comes your T cell. When this T cell doesn't see a lot of S1P, it kind of misses it, okay? So that T cell, it starts to produce a receptor on the surface that is specific for S1P, okay? And it's really looking for S1P because it doesn't see a lot of it, okay? So without seeing a lot of S1P, the T cell is going to produce a lot of this receptor because it realizes that it likes to interact with S1P, okay? So as it is getting closer to a site where you have more and more S1P, so now this T cell is saying, okay, I think I interacted enough with it. I'm kind of bored now. I don't want to interact with it that much. So when it starts to see more and more S1P, the T cell starts to downregulate the receptor. So this T cell guy is kind of undecided, you know. When it doesn't have the interacting thing, it wants to interact with it. When it has it, it doesn't want it anymore. You know what I mean? Yeah, I mean, the cells have issues, like some human beings. But anyway, get that in your mind, okay? This is like a complicated relationship. So, how does that work? Exactly as it does with human beings. Like, I miss you, honey. Can you just come to me? And it's like, well, make more of your receptor. Sure, I'll just make more receptors. Please interact with me. No, oh, come on. Okay, so you get to a point that you have a lot of S1P. Okay, let's interact a lot. And then you're like, I'm tired of interacting with you. Let me downregulate my receptors here. I'm not going to be responding to you as much. But now it's like, oh, but now I'm really into you. Now I got a lot of receptors here. I mean, we got to do something. And guess what the T cell does? It downregulates the receptors even more. It's totally a turn off for the T cell. Literally, it turned off the receptor expression. Okay, now the T cell is like, you're clinging to me, I don't like this. So what is it doing here? is going to extravasate through the high endothelial venule and is going to jump into the node, into your lymph node. Escape strategy. Okay, and what's going to happen in the lymph node? Look at this. There is not a lot of S1P in the lymph node. And what's going to happen to the T cell that doesn't have receptors now? Okay, who's going to guess? What's going to happen? It's going to start missing S1P again. It's going to make more receptors. I told you it's complicated, right? The T cell here, when it's in the lymph node, looking for the pathogen, and let's say it doesn't find it, right? And it's like, you know what? It was kind of nice interacting with the S1P guy. I kind of miss him. You know what? Let me start making some more receptors. Okay, a little bit more receptors, because there is not a lot of S1P around. And then what's going to happen? With this whole flow that is going through the lymph node, the T cell is not going to be sitting there. It's going to go with the flow. It's being filtered, and then it's going to come out of the lymph node through the efferent vessel. And look what happens here. At the efferent vessel, you're going to have a lot of the S1P molecule again. And now your T cell is full of receptors again. And what's it going to do? Oh, S1P, I missed you. And it's going to go towards there. And it's going to interact with it again. And with the flow of fluid out of the lymph node, it's going to be pushed out of there. Does that make sense? So it's not going to hang around the lymph node forever. Okay? So you'll be pushed out of there. And then it gets to the lymph. It's going to, it's coming out here from the lymph. It's going to get to the circulation. And then it's going to get to the blood again. Okay, towards the blood recirculation. Is the story making sense for how this mechanism works? Okay, everybody cool with that? Okay, just put in the context of, unhappy. They're gonna, go into this secondary conformation. And when you look at those small proteins, or those cytokines, you will see that to hold the secondary structure together, a lot of times they have covalent bonds between the amino acids, right? And this one amino acid that is the only one that is very important for covalent bond formation is cysteine, because cysteine is going to allow you to make disulfide bonds. So everybody took a molecular cell? No? Yeah. Biochemistry, molecular cell, you know what I'm talking about? Cysteine bridges, cysteine double bonds. Okay, so when you look at these proteins and you say, oh, let me look at the primary sequence of amino acids, and let me look at how many sulfur bridges you have, right? Disulfide bridges you have, and then I'm going to group those proteins into different families. So the first one that you see here, you're going to find two cysteines that are interacting with each other and making this one bridge, one disulfide bridge. So you're going to call this a C family cytokine. Okay, so this one, for example, is called XCL1, but you have XCL2 or 3, meaning that the X would be another amino acid on the sides. But here you're going to have basically one disulfide bridge of cysteines that is forming. So that is your C family of cytokines. Now, if instead of having only one disulfide bridge, you're going to have two disulfide bridges, and your cysteines come side to side, they're very close to each other, right? They don't have anything in between, then you call that a C family, because C is a cysteine. So cysteine close to cysteine is your C family. Okay, so here's only one cysteine in that sequence. Here are two cysteines, so that's your C family. Now, you're going to find, cytokines, one of the most important for that family is called RANTES. Okay, it's also called CCL5. So that one is very important for inflammatory process and antiviral activity. Okay, so this is your CC family of cytokines. Then you look in other proteins and you're going to say, okay, I have 2 disulfide bridges, I have 2 cysteines, but now you have this other amino acid in the middle. And like, which one? It doesn't matter. Anyone. So we call it X. Okay, which one is there? Anything but cysteine. Okay, so if you have an X in the middle, 2 disulfide bridges, you call this family CXC family. That makes sense, right? CXC family, all right. And by the same token, if you look at the protein and you have 3 amino acids between those cysteines, you're going to call that CX3C family. Okay? At least they didn't call it CXXXC family. Imagine to name them, yes.
Is it like a structural thing, or is it possible to just have 2?
Oh, exactly. Why they don't have only 2? Well, they don't have a CX2C. I never thought about that. I think they could. For some reason, they don't. Maybe it is a hindrance type of system, but I never looked into it. Okay, so you don't have a CX2C family. Okay, that was abandoned. Okay, so you're going to have a C, a CC, CXC, or CX3C family of cytokines. Okay, and when you get to this complicated family here, the names get even more confusing because it's going to have a common name and a formal name, and that's when you start getting a lot of letters and numbers. Anyway, we're going to get there. Of course, you're going to have to know the very important ones, like, for example, CXCL8. That is your interleukin 8. And remember what that's important for? It's an inflammatory cytokine, mainly. Okay? All right. So everybody cool with this naming of families? So, how is it the cytokines are working? They are going to be attracting cells depending on the gradient of those proteins. So if you have a little bit of proteins here, then your T cell here, or your lymphocyte, is going to be attracted a little bit, but then it wants more. So it's going to go down that trail to where it can find more and more cytokines present. Okay? So, I mean, down the trail, but it's really up the concentration gradient. The more cytokines there are, the more attracted the lymphocytes are going to be to this particular site. Okay, so they go up the concentration gradient. Okay. So, cytokines are the molecules, right? The little proteins. And how is it that they're going to interact with the cell? Well, the cell has to have a receptor for those molecules. So the lymphocytes have these receptors on their surface, okay? And this type of receptor is called a G protein coupled receptor. Has everybody seen that before? Who has never seen a G protein coupled receptor? Okay, very important for signal transduction. Okay, this type of receptor is not going to allow the molecule that is interacting with it, is not going to allow the molecule to go inside, but instead is going to transfer the message from the outside of the cell to the inside of the cell by this communication or mechanism that involves G proteins. Okay, so that's your signal transduction. That's subject for a different class. I mean, different class altogether. Okay, molecular biology. Okay, but so these receptors here, what is important thing you should know? They're G protein coupled, so they're together with G protein that is on the inside of the cell. These receptors are the 7 transmembrane spanning receptors. So they have 7 transmembrane spanning domains, and if you count them, you go 1, 2, 3, 4, 5, 6, 7. That's what it means. Okay, it goes across the membrane 7 times, and they're really going to transduce the signal to the inside of the cell, and that is going to tell the cell what to do, what genes to upregulate, what genes to downregulate, and how the cell has to behave afterwards. Okay, so the nature of this chemokine It's very important to reach the right receptor so the cell knows exactly what it has to do afterwards. Okay? Everybody cool? All right. Okay, so here. Characteristic or personality of your T cells that changes, okay? So let's see what happens here. When you have your naive T cells, at the surface of the naive T cells, of course, you're going to have all different types of receptors, right? But at the surface of your T cells, you're going to have that very important molecule that is going to allow it to keep on circulating, okay? And that is your CCR7. So these guys that had never seen antigen before, so pay attention because this is the part that gets a little complicated and usually flies off your head, like, ooh, what was she saying? CCR7, CCR8, is that a song? Okay, that's usually what happens, I tell you, okay? So look at this, naive guy, never seen antigen. It has to circulate. So it makes sense that it has something on the surface that is going to allow it to circulate, and that's your CCR7, okay? CD62 is also important for that, but I didn't even mention that. So you focus on CCR7, okay? Now, once the cell has seen antigen and it needs to become a certain type, it needs to keep the memory of something that it has seen, right? That's the whole idea for your adaptive immune response. You always have memory of stuff, right? So cell sees antigen. Instead of becoming one of the army fighters, it is going to become the general that remembers the fight, okay? So if it stays in the lymphoid tissues and is the general that remembers the fight, so now it's going to be differentiated into some type of memory cell. Okay. Now, this memory cell is going to be hanging around the lymphoid organs. Okay, so in this case, you're going to call this memory cell a central memory cell. Okay, so let's see what is the difference between a central memory cell and a naive memory cell. There is only one difference in the receptor here that has to do with the CD45RA. Okay, and RO. Okay, RA and RO, they go together here. Once it has seen antigen, it's going to flip the expression of those receptors. Okay, but now this guy is not going to be a fighter. He's not going to be a mano a mano. He's going to be just the guy remembering. Okay, so what's going to happen here? The guy wants to patrol the lymphatic organs, secondary, of course, right? You want to patrol the lymphatic organs. It wants to have a memory of what it has seen. So it's going to look around and it's going to be ready to get activated and make more soldiers if it needs to. So that's going to be a fast secondary response. Okay. So what happens here? You have some receptors that were switched in the expression, but you keep your CCR7 expression because that guy is patrolling around. Okay, you want to keep on moving from side to side to find the fight. Okay, so this is going to be a characteristic of your central T cell memory. Okay, T central memory cell. Okay, now so this is happening in the lymphoid tissues. Okay, when the T cell is a effector T cell and it leaves the lymphoid tissues, it gets to the periphery. So now you have a cell in the peripheral circulation, okay, and the cell is an effector cell. It has differentiated. It knows the job that it has to do, okay. So something else is going to happen. First thing that is going to happen here is the CCR7 is not going to be there anymore, because now this cell doesn't want to go back to the lymph nodes. It wants to go to the site of battle. It has to change its course. It's useless if it keeps on going back to lymph nodes because it already found the antigen. So it has to go to the tissue where the infection is happening. And then after it fights the infection, what's going to happen to it? Is it going to find its way back to the lymph nodes? Okay, if you have to guess on something, what's going to happen to it? All right, let's think medieval war. Braveheart, anybody watched it? No? I mean, any medieval wars that you guys can have in mind? Did you watch those movies? Okay, what happens in Braveheart? You have all these guys that first they were just commoners, they were naive, and then they became soldiers, they got painted, right? And they go to the battle and they're like, hey, it's over there, fight! And they go fight. What happens to most of them? They die. Gruesome, awful deaths, right? They explode, they get things that come from the inside. No, that's another movie. Okay, they die, generally, right? The drama. So these cells here, they don't even have the receptors. If they wanted to go back to the lymph nodes, they know they're going to die. If they don't die, that's a problem, right? You're going to see that later. If they don't die, that's chronic type of stuff, okay? But look at this. Your effector T cell does not have CCR7, okay? Now, your effector memory cell that's going to be patrolling around also doesn't have CCR7. So CCR7 is a good thing to keep in mind, that is going to tell you the stage of development and differentiation of those cells, okay? As well as CD45RA and CD45RO, okay? When you are in the periphery, you are also going to have the type of configuration of those two receptors that you have when the cell was naive, but the rest of it is going to be different. Okay, so make sure you go over these receptors here in a way that is going to make sense to you. Okay, you know the differences here. Okay, so here, yeah, little things that are emphasizing here. Different receptors depending on what it needs to interact with. So here is a bit more on the description of your adhesion molecules that we had mentioned before, right? That you have those CAMs, they're cell adhesion molecules. You have several types of those CAMs. And here is just telling you, you know, different sugars that they can interact with. So that's very much just FYI. Okay. And this is the part that gets really, really interesting. Okay. When you have activation of those receptors on your cell, when you have interaction of things the cells have to interact when they get to a certain site, okay, those lymphocytes, okay, they can activate receptors in two different ways. Okay, this is the part that gets also a bit confusing. Okay, so if you look up here, they have some receptors on their surface that are inactive. So they are in a low affinity conformation. They are bent to the side. Active side of the, or binding side of this is not exposed out there to interact with anything. Okay. So there are two ways of activating this. One way is that the cell can send a signal from the inside of the cell. Okay, remember when the cell goes to like extravasation, goes to a place, the cell is changing the shape, is modifying its actin cytoskeleton, right? Is modifying its network. And if the cell is really pushing to go to a certain area of infection, it is going to upregulate this molecule from the inside that is called tallin. So tallin is going to interact with this actin pushing and pulling network from the inside of the cell, and is going to get to the inside part of this receptor and is going to start interacting with it. Okay? Once tallin interacts with this receptor, it's going to change the conformation of the outside of that receptor. Okay? So this type of signaling to change the conformation of the outside of the receptor from the inside, and now this thing is going to be able to interact with something else that is supposed to, this is called inside-out signaling, which is very descriptive and should make sense. You are getting the signal from the inside of the cell, so it's inside-out signaling. Does that make sense? Yeah, I see people are getting ready to leave. Okay, we just one more minute I'll take here. So if I mention an inside-out signaling, you can imagine there's going to be an outside-in signaling, right? So here is another type of example of an outside-in signaling, where a lot of ligand gets together and catches that conformation when it's about to be receptive to ligand activation. Okay, so we stop here with the idea of inside-out and outside-in signaling. We'll pick up from here next time.